now welcome back, and we would finish the second part of our lab by finishing the t-SNE analysis. So remember that in the first part, we have already clustered all the cells into different groups by running the t-SNE, and now the last job we want to do is just annotate all the groups into different cell types. For example, you can see that in this t-SNE plot, you might have group 0 to 7, but you didn't know which group refers to what type of cells. This plot might be something you don't want it because you can't find any information for you. And if you go back to our lecture, so what we have done now, we have already visualized the raw data and do some quality control for the raw data, the normalization, the visualization, and then run the clustering for the linear dimensional reduction method, such as the PCA. And now we are doing the non-linear dimensional reduction method by using the t-SNE. And the last job is to annotate them. So now we want to annotate the clusters. And how to do so? The first step might be find some statistically significant identifiers, which we call them as cluster biomarkers for each cluster. So these are actually some I would say that as signature genes. So these genes might come from some specific cell types, which means they expressed differently in this type of cells compared with all other cell types. So first, we have to find these biomarkers, and then by using the biomarkers as the bridge, we could match all our clusters to the target cell type. So to find the markers, the package provided a very easy command as the find marker command. So by setting the threshold of the identity.1 equals to the cluster that you want to find the that you want to find the signature biomarkers here. This means find all the biomarkers of the cluster 2, and if you want to do some comparison between different clusters, you can set an additional parameter in this command as the identifying.2. So basically, this command just find all the biomarkers that could distinguish cluster 2 from cluster 4. You can also find biomarkers that could distinguish cluster 2 from a cluster like 1 and 4, and do any combination what you want. So just change the command in the identity.2. But I don't think you might need the second parameter for your homework. So for your homework, actually what you need is just the identity.1. So you can change it as 1 to 7, 0 to 7. So remember, you have got 8 clusters, and you can find all the biomarkers that could identify these clusters from the remaining part by using the find markers command. And so simply use find all markers. You can also do this one by use this command I provided here. So you can find markers for every cluster compared to all the remaining cells. And now we might report the top 2 genes that could identify this cluster. So this command could provide a table of this strategy. So now from this table, you can see that for cluster 0, actually the top 2 biomarker identifiers might be gene LDHB and gene IL7R. And then same for cluster 1, the top 2 significant biomarkers might be considered as S188 and S189. And for cluster 2, the top 2 biomarkers might be CD179A and TCL1A. So by using the feature plot, so this is just a literature information. If you want to see some more intuitive ones, I would recommend you guys to use the feature plot to visualize them intuitively. For example, if you for example, yeah, so here I have already provided all the feature plot for all these top biomarkers. For example, if you remember the biomarkers for cluster 0, they are LDHB and IL7R. So if you check the LDHB and IL7R in the feature plot, you can see that all these cells are clustered into the group in the center. So which are this group? Actually, it's group 0. So which means they identified the first cluster 0. So and similar things happen to IL7R. They are also grouped into cluster 0. And for S188, according to our literature information, it's basically from cluster 1. So if you go back to your t-SNE part, you can see that they were accumulated distributed around cluster 1. You can do the same thing for all the remaining parts. So by using the feature plot, you could see you could match your clusters in your t-SNE results to the significant biomarkers. And the last step is to okay, this is another method to help you guys to match the clusters to the genes. For example, by you can plot the violin plot of the genes. For example, if you check the CD79A, remember that they are the sorry, yeah, here. Remember that the CD79A actually are the biomarkers of cluster 2. So you can see that the distribution of cluster 2 behaved very abnormal compared to the remaining clusters. And the LST1, if you go back to check the table, so actually they are the biomarkers for cluster 4. So in this violin plot, you can see the distribution for this the fourth cluster uh, behaves very strangely compared to all the remaining clusters. You can do the same thing for the remaining genes. So all these plots are actually providing you information about how to match the genes to how to match the biomarkers to the groups you find in in your t-SNE plot. You can also use the heat map might be quite busy, but it could provide you a lot of information. So this is this is very busy. Actually, it provides you all the top 10 genes within each cluster that could identify all the biomarkers. For example, in the cluster 0, you can find the top 10 most important biomarkers, and so for cluster 1, 2 to 7. And you could use all these biomarkers to see what kind of cell are actually referred to this cluster. So that's our last step. So remember now you have already matched the biomarkers to your clusters, but the last step is to match the biomarkers to some specific cell type. So this step might require some literature review, but I have already done something for you. You can check this table. So for example, you can see that the IL7R markers might be related to the CD4 T cells, which means also remember that IL7R actually is the biomarker for cluster 0. So which means that you might want to annotate the cluster 0 as the CD4 T cells, which is actually the central part in the t-SNE plot. And so for the remaining part, for example, the CDI4, let's see whether they are biomarkers. Oh, this is not a biomarker. Maybe we could do something about this. Yes, you can do the remaining part for all the other biomarkers to see which cluster refer to like either one of these 8 cell types. And you could do some further literature to see whether IL7R is related to CD4 T cells or the S188 is related to like either one of this cell type. And you have to fill in your cluster ID in your t-SNE here. So this is for question. Let's see what question is that. It's for question 10. Yes, I think so. All right, and now finally we can annotate the t-SNE groups now. So basically, you remember that the original setting is from 0 to 7. You actually just change the cluster IDs from 0 to 7 and some specific cell types here. So then you could see all the annotation beside your plot. All right. If you go back to our lecture, you can see that Great, we have already completed all the tasks we might need to do for a typical single-cell sequencing data analysis. And if you guys like to go through all the questions one by one, you can follow me. And if not, just stop here. And so for this homework, we'll be analyzing a single-cell RNA sequencing dataset of the PBMC from the 10x genomic platform. Well, same thing for the not the same data, not the same original data, but similar things for our lab. You can see that there are more samples compared to our labs. So, you don't need to check the original dataset. I have already provided them in the GitHub for you guys. So basically, it's some zip folder. If you download it, so it's this file, and you unzip them, you might get a folder. And by click them, another folder, and click it again. So this is the most updated version that the Seurat group adopted for their data. So actually, so originally, remember that they store their data in just one .rds file, but now maybe they want to save some storage for their data, so they separated them into two separate files. So the result is that you have to change the way you input your data in the homework. So now you have to use the read 10x command, which is developed by them recently, to read all the files under this folder. So actually, we're reading all these three files into our R session here by using the read 10x file. But now, if you check the data structure, they changed a lot. For example, if we log in the R session for this lab, I can show you from the R. Alright, the first thing is to library the required packages. So this read RDS is our original strategy for input the old data type. And now if you go to the homework. Alright, so if you read in the 10x, or use the read 10x command, you can see that now the PBMC, the PBMC.data actually didn't contain the raw data, the data, and the scale data. They contain something quite different. So that's the new data structure, which is quite different from our lab material. So, but you can use the dimension command to visualize the dimensions of the data now very intuitively. So you can also print out the number of the genes and number of cells here. And to calculate the dropout rate, actually it's asking you to calculate all the data that is equal to 0 and sum up all their numbers. If you simply type this command, so you can get around this large number of subjects with data equal to 0. And if you want to calculate the ratio, you might use divided them over the multiply results of the dimension. So now you would get the ratio as around 97%, so which is very high because the original dataset is really sparse. Alright, that's all for the first question. And similar things happen to the second question. But for second question, what we were doing is to create a new Seurat object, as I mentioned, by filter out some problematic genes and problematic cells. But if you do this step, and you created a new data structure, which is actually the Seurat data structure. Yes, for now you have already got raw data, and scale data, which is the original setting for the typical Seurat object. So now you can do all the operations I have done in the lab. Like you can view the dimension of the PBMC data at raw. Now, yes, now it worked. All right, sweet. And for the third question, actually it's doing some quality control steps. So they are all commands are on the QC slide. So I want to make some corrections for something I did in the lab. So for this new data structure, you might need some additional commands for the QC steps. I give all the hints in the homework, and you could do the same thing for your task, and then could visualize them by generating the violin plot and using the filter cells command to filter cells with extremely high coverage. Then for question 4 is asking you guys to run the PCA. Remember that we have to do some normalization before we run PCA, and then you could find the variable genes across all the original data, and then you have to regress the noises out from your variable genes by using the scale data command. So the potential confounders we used here is the number of UMI and the percentage of mito genes. The last question is asking you to see how much contributions does each of the PC contributed. So you have to print out the eigenvalues here. You might need them to calculate the variance that could be explained by all the PCs. So it would be fine if you calculated the variance contributed by each of the PCs, but I would suggest that the variance contributed by all the PCs might be more informative because you are using the total PCs to do the clustering issue. And for question 5, I have for graduate students, I have give you a lot of hints here. And if you run this code, you might find some really interesting results. Just try to explain them as much as you can. So for question 6, it's asking you to do the t-SNE. So 6 to 10 are all doing the t-SNE. So first, you have to find the clusters. It's basically doing in the find cluster command, and then run the t-SNE, and then compare the t-SNE plot to the PCA plot. So the PCA plot actually adopted the clustering results from the t-SNE. So actually, you will plot the same clusters of t-SNE into the PCA, just trying to say whether whether these groups are separated very clear in the PCA plot. And for question 7, you are trying to check with different seed. So this command is not correct. You might consider using the seed.use command to change the seed while you are running t-SNE. And for question 8, is doing the same. Question 8 is doing the similar thing compared to question 7. What's different is just changing the resolution. So it's in the same command as the run t-SNE. In run t-SNE. And for question 9, it's visualized the gene expression. So it's asking you to find all significant markers for each cluster. Just use the find all markers function and do the feature plot to see to match the cluster to the biomarker. And for question 10, just match the cluster to some specific type of cells through the biomarkers and identify each cluster. All right, and up to now, you have finished all your homework. And if you have questions, please come to my office hours and ask me about all of them. And I will hold an additional office hour on each Friday since this week, except the spring recession, and also the open house week on the end of March. So good luck for you guys for the remaining part of your homework. I will see you in our next lab. Bye.